We have our section where we're now going to compare the various types of muscle tissues. We want to introduce, of course, now cardiac muscle, unique to the structure of the heart, the four chambers, responsible for our blood pressure, pumping our blood. It has this involuntary control via the nervous system. So we do not have that direct conscious control of this type of muscle tissue. The same applies for smooth muscle that's found in various hollow organs, open tubes, such as arteries and veins, the digestive organs, the stomach, the intestines, the uterus, the iris, all great examples. And going back to chapter 3, of course, just to start for the comparison of the three tissues, we have locations, of course, for each. Cardiac muscle tissue has this unique type of cell junction connecting one to another, this intercalated disc, and an image of the histology, a sketch. And taking a look at all three, we have the long cylindrical shape for skeletal muscle, multiple nuclei in one cell, the branching fiber shape of cardiac muscle, and this spindle tapering down shape for smooth muscle. Here's a good representation. Lacking any striations. We've seen the histology of cardiac muscle. Intercalated discs present, unique to this one. Smooth muscle, lacking striations. Here's one view of skeletal muscle, striations with multiple nuclei at the periphery, or another color stain treatment of skeletal muscle. And this led us to the study of the sarcomere. So again, leading to that organization of I bands and A bands alternating with the sarcomere arrangement, which is present for and responsible for making the striated appearance. So we have these 10 traits listed here, and we'll just go through and do a comparison of the three types. So starting with the location, cardiac muscle unique to the heart, no other organ, so that exclusive location. Smooth muscle, we just mentioned a number of hollow tubes, and in the wall of those hollow organs, there's a layer of smooth muscle, arteries, veins, the uterus, all great examples. And skeletal muscle, of course, the muscle tissue making up our muscular system, so the specific muscle organs, the biceps and the hamstrings. Looking at a single cell, the number of nuclei per individual cell. The usual case for cardiac muscle is 1, and a rare case, some do develop 2 nuclei per cell. Smooth muscle are uninucleate, there's only 1 nucleus per cell. Skeletal muscle is multinucleated with dozens of nuclei per 1 skeletal muscle fiber. The shape of the cell, so we have a description here, the branching fiber, the fact that one or more ends of the cardiac muscle cell branches and splits, the tapering spindle shape, or a long cylinder for skeletal muscle. And we could just go back to the sketches for that. So the long cylindrical shape for skeletal muscle. Here we see that branching fiber structure on cardiac muscle. And here's now that spindle, thicker in the center, tapering to the 2 ends for the appearance of smooth muscle. Back to our chart. So now we have these two which go hand in hand. If there is a sarcomere arrangement, the alternating A and I bands are the reason why we see these striations. So we can tackle these together. Cardiac muscle, Well, when there's a sarcomere arrangement, it'll have striations. Smooth muscle lacking the sarcomere arrangement means that it lacks striations. It does not have the organized A and I bands. Skeletal muscle, again, contains the sarcomere structure and thus has the appearance of striations. And that's coming from this here. Here's the sarcomere arrangement. And we have the arrangement of light and dark bands repeating. That gives us the light band, the dark band, the light band repeating over, making that striated appearance. So now we have these two types of cell junctions. Intercalated disc, of course, unique to cardiac muscle tissue. The other two types of muscle tissue are lacking intercalated disc with strong physical connections between cells. Gap junctions, another type of cellular junction. In this case, it not only bonds the cells together, but there's also this direct ion channel between the cells. And that's truly its purpose, is an ion channel. So this is also present in cardiac muscle tissue, as well as we notice here, smooth muscle. However, skeletal muscle lacks the gap junction ion channel. Now starting to look at how these are typically now being stimulated. So these last 3 points here, the voluntary conscious control by the nervous system. It's not present for cardiac or smooth. So we can't at will decide that we want just simply our heart to beat faster. If you're sitting in a chair, you can't decide that you want your pupil to dilate. There has to be some other influence, and we get that indirect involuntary function for those two. Whereas skeletal muscle has both. You could decide at any point you want to wave your hand, you have that voluntary conscious motor control. But also, a great example of involuntary function is your diaphragm. You're breathing, even when you're asleep unconscious, you can still contract that skeletal muscle. And the final one we want to tackle now, just typical stimulus to contract for each type. In the heart, there's the 2 pacemaker groups that stimulate the contraction of cardiac muscle tissue, typically. There's many other sources. For smooth muscle, a major source is simply stretching of the cell, stretching of the tissue. So you think, for example, filling your stomach, expanding the wall of the stomach and the stretch on that muscle tissue. And the response is it'll begin to contract. And truly, this applies to all 3 types of muscle tissue. Stretching a muscle cell stimulates contraction. And skeletal muscle, of course, we've seen the neuromuscular junction, our nerve cell releasing ACH to stimulate our skeletal muscle fibers.